Welcome back. We are now going to discuss further about the molecular mechanisms of the carcinogenesis. But before we move ahead, let's briefly review what we have seen already in our previous lecture. In the previous lecture, we had already discussed about the RAS, MYC, and the cyclin D and the cyclin dependent kinase inhibitors. And recall all of these were the proto-oncogenes, and when they were converted to their oncogenic form, they would lead to cancerous conditions. Now, we had also discussed about the Darwinian selections. The Darwinian selections, which were important to understand why some tumors are difficult to treat because they acquire tons and tons of mutation as the tumor progresses. And the Darwinian mutations are also important because the cancer cells evade the host response, which we are going to see further in this lecture. Okay. And as you can see, we have discussed only the one out of the four mechanisms, and we are going to discuss the remaining three now. So first we'll see the tumor suppressor genes. As the name indicates, these genes help in the suppression of the tumor, and the most important of them are the p53 protein encoded by the TP53 gene, and the second one is the retinoblastoma, that retinoblastoma gene. Okay. Now, two-hit hypothesis, this is something that we need to understand before we go further to understand the p53 and the RB gene pathways and mechanisms. Now, we know that there are two alleles of a gene, and the two-hit hypothesis simply states that both the alleles must be damaged or they may be defective. So, this that would lead ultimately to increased risk of cancer. And as I've stated that normally mutations in one gene, only one gene, that is one allele, would rarely produce a neoplasm, while in the familial cases, one defective copy is already present and the other copy that is damaged because of some spontaneous somatic mutation. So that would lead to increased risk of cancer. So the idea here is that familial cases would have increased risk of cancer because they already have one defective copy of the allele, while in a normal person there are somatic mutations, but they only affect only one allele, and the other gene or the other allele remains patent or it remains normal, and hence they rarely have a neoplasm. Now we are going to discuss about the RB gene and the P53 pathways and mechanisms. But first we need to understand that retinoblastoma and P53 are also the part of the regulation of the cell cycle. So as I've already mentioned in the previous lecture, the cyclins are the promoter. You can remember by cyclins causing cycling of the cell cycle. The CDKIs obviously would be the inhibitors because cyclin dependent kinase inhibitors, and they cause decrease in the cell cycle. Now we have the retinoblastoma, that is the RB gene, that is the inhibitor. When the RB gene is activated, as we will see in the lecture ahead, that RB gene is an inhibitor of the cell cycle, and P53 also is a inhibitor. Now the retinoblastoma, as I've mentioned previously, that in the two-hit hypothesis, uh, the normal cases of the retinoblastoma type of cancer is not seen sporadically because both the alleles must be damaged, and therefore the retinoblastoma type of the tumor is seen mostly in the familial cases. The second thing to know is that P53, some authors refer to it as the guardian of the genome. Now we are going to see the retinoblastoma and its mechanisms, but let's draw a broad picture first, and for that let me draw a whiteboard. So let us connect the points, the cyclins, the retinoblastoma activity. So say we have the growth factor that stimulates the cyclins in the cell. Now when the cyclins are stimulated, they cause the decreased RB activity, and that is done by the hyperphosphorylation. Now when the RB is hyperphosphorylated, its activity decreases, and this causes the freeing. The E2F is normally bound by the retinoblastoma. Actually, let me draw it in the side. Let's say we have this E2F, and this is the E2F molecule, and the retinoblastoma just holds the E2F like this, as I've drawn. So when the retinoblastoma is phosphorylated, this E2F escapes, and the E2F is the elongation. It is a transcription factor, and it causes the transcription to increase, and this causes the cell cycle to proceed. Now imagine what would happen if we have the growth inhibitors like TGF beta, or growth inhibitors, as I've already mentioned, from the growth inhibition signals from P53. So that would cause the CDKI inhibitor, that is cyclin dependent kinase inhibitors, and this would cause hypophosphorylation of the RB gene, that would cause its activity to increase, and this would in turn cause the E2F to be more bound and to be more hidden, that is because of the RB activity increase, and this would actually block the transcription and the cell cycle won't proceed further. So this is the basic idea behind the retinoblastoma and its connection with the P53, the cyclins, and the CDKIs. So let us see the slide now. So as I've already explained, the retinoblastoma gene helps the transcription to block with the help of the E2F, and that it blocks the E2F, that is, it holds the E2F when it is in the hypophosphorylated state and its activity increases. As I've already stated, that retinoblastoma is a tumor suppressor gene, and it suppresses the cell cycle. So when in the hypophosphorylated state, again it is active in the hypophosphorylated state, and this is the sort of the flow chart or the flow diagram that I've already explained, that when the cyclin D are active because of the growth promoters, it causes the hyperphosphorylation of the retinoblastoma gene, which causes the E2F to become free, and the transcription occurs, and it removes the inhibition at G1S phase. Then the retinoblastoma inhibits the E2F, which as I've already stated, that it is a transcription factor that causes progression from the S phase. Now, who regulates the retinoblastoma gene activity? Now, as we have seen in the flow chart, that before RB comes the cyclin D or the CDKIs, and after RB comes E2F. So we can think easily that if the cyclins, that cyclin causes cell cycle to move ahead and to progress. Therefore, if cyclin is active, then obviously the retinoblastoma must inactivate itself, and that is when it is in hyperphosphorylated state. The second thing, with this only we can understand that when cyclins are inhibited by the CDKIs, the cyclin dependent kinase inhibitors, it would in turn cause the RB activation, and P53, as I've already stated previously, that P53 activates the some cyclin dependent kinases that activate the retinoblastoma, increases its activity and prevents the cell cycle to proceed further. Now it all again depends on the E2F activity, that whenever the RB activity is increased, the E2F free E2F decreases, and that leads to blockage of the cell cycle at the G1S phase, while if the E2F becomes active and it is free, the cell cycle proceeds. Now the general concept would be that in the whenever we see there is mutation in the retinoblastoma, we can say there is cancer. Obviously, the mutation should be in both the alleles. But now, if there is mutation in upstream or downstream, upstream means in the cyclins or in the CDKIs that, and also in some growth promoters or in the growth receptors, or even the P53, that is the inhibitor, they would indirectly affect the activity of the retinoblastoma gene, and therefore we can think that it is upstream or downstream, whether E2F is also dysfunctional or it is overfunctional, then all these things would cause the problems and cancer to occur. Now, the acquired mutations of the retinoblastoma gene behave in a recessive fashion, while the congenital behave in the autosomal dominant form. And we can understand this point with the two-hit hypothesis easily, that let's say we have these two alleles, and the genes are there, two alleles of a gene, that is the RB gene we are talking. And if during the somatic mutations, one of them is defective, And now if this mutation has to be transmitted, this this would be a recessive kind of mutation. Why? Because there would be no cancer, that is the retinoblast, there would be no retinoblastoma cancer, and that's because both the genes must be damaged to express the cancer phenotype. But let's say we have this another type in which the familial form, in which the one gene is congenitally familial form, and one gene is congenitally defective. Now, with the help of the somatic mutations, the another gene would also get damaged, and that would lead to the retinoblastoma phenotype, that is the retinoblastoma cancer. And so we say that if one gene is defective, then ultimately, or obviously, it's obvious because of the somatic mutations that normally occur, it would be have in a dominant form. Okay. Now we are going to see about the TP53, the tumor suppressor gene. Now, what are the normal functions of the P53 protein? So it arrests the cell cycle. I've already said that it is the guardian, and it protects the cell. So it arrests the cell cycle if it sees there is some damage. And why it arrests the cell cycle? Because there may be some DNA damage, and for it may cause DNA repair. And if the DNA repair is beyond the scope of repair, it would induce senescence or apoptosis. So it's easy to remember the functions. Now, the sporadic cancers have the P53 mutations, and as I've already stated, that mutations are not limited to the tumor suppressor gene itself, but also to the upstream and the downstream pathways. Let's say the retinoblastoma gene is defective, then that would appear as the P53 damaged P53. So just keep this in the back of your mind that there may be some problem in the downstream or upstream pathway also. Okay. Now, how the P53 arrests the cell cycle? Now, I have already explained this, but let us go through it once again quickly. P53 arrests the cell cycle by maintaining the retinoblastoma gene in the active state. And I can stop here and ask you in which state, in the hyper or hypophosphorylated state, the RB is active. Excellent. I actually have already mentioned over here that in the hypophosphorylated state, the retinoblastoma gene is more active. Now, if the P53 is active, that would cause the cyclin-dependent kinase inhibitors to be active, and it would cause the RB in active state, and then the E2F activity should decrease and inhibition of the cell cycle. Now, there is some one CDK which is directly dependent on the P53, and that is CDKN1A. Okay. Um, uh, Ahead in the lecture, I would tell you about the CDKN2A that is involved in the functioning of RB as well as the P53. Okay, so till then keep this thing in your mind that CDKN1 is only the P53 dependent cyclin dependent kinase inhibitor. Okay. Now I have mentioned the four function of the P53, and we have seen the about the cell cycle arrest function. So what about the DNA repair functions? The DNA repair is with the help of the GAD45, and this is something that you need to remember. Probably a low yield point, but still I've included for the sake of completeness. Now, the P53 causes apoptosis, and that is mainly by the intrinsic mitochondrial pathway. High yield principle there. Intrinsic mitochondrial pathway, and that too by using the BAX and the PUMA. Recall that these are the pro-apoptotic molecules, and the BAX and all would cause the decreased BCL2 expression, and that would lead to apoptosis. Now, senescence is also induced by the P53, and that mechanism is quite not known, but it is believed to be because of the epigenetic silencing. Now, as we have seen in the RB gene pathway that there were something that used to dictate its activity. Those were the P53, the cyclin dependent kinase inhibitors. And so is the case with the P53, that it is negatively regulated by a thing known as MDM2. MDM2 is an inhibitor of the P53 protein. Now here comes the CDKN2A, high yield. What is the function of 1A? Let me just say something over here, that P53 activates the CDKN1A to arrest the cell cycle, and CDKN2A activates the P53 pathway to arrest the cell cycle. So the function is same, the goal is same, that is the CDKNs must inhibit the cell cycle because they are cyclin dependent kinase inhibitors. The goal is same, but only that CDKN2A activates the P53 pathway while P53 activates the CDKN1A. Okay, and CDKN2A activates the P53 pathway by blocking the MDM2. The second thing to understand is that there are some gene, the ATM ATR gene, the full form would be the ataxia telangiectasia mutated gene, or the RAT3 mutated ones, sense the DNA damage and they activate the P53 pathway again by inhibiting our MDM2. Okay, so high yield to know that MDM2 is negative regulator of the P53, and the CDKN2A promotes the P53 pathway by inhibiting it. Very high yield to know that HPV, human papillomavirus E7, inhibits the RB gene. Very high yield to know. And the HPV E6 inhibits P53, again a high yield concept. Now E6 is also associated strongly with the cervical carcinoma, and that is the thing that we check with the cervical dysplasia in the pap smear. Okay. Now, as we have seen in the retinoblastoma gene, that the people who have both the alleles defective are predisposed or have retinoblastoma type of tumor in their life, and also having this risk of tumors, other types of tumors. And the same here in the when the TP53 allele that produces the P53 protein is defective, one of the allele is defective, it leads to a syndrome called Li-Fraumeni syndrome, and it presents with cancers at a young age, and these people are 25 times more likely to have cancer by the age of 50. Now here I have included the list of the tumor suppressor genes that are most important, and what happens when they are present defective familially, and their mechanisms. Okay, so the CDKN2A, as I've already stated, governs the activity of both RB and P53, and it is inhibitor, cyclin dependent kinase inhibitor, so of course it would cause the cell cycle arrest or the inhibition of the cell cycle. Now it is very high to know that the mutation in the CDKN2A, and also we can see the P53, because it is the upstream pathway, CDKN2 is the upstream pathway of the P53, is linked to melanoma, familial melanoma. This is something which you should remember. It appears on examinations. Okay. Now CDKN familial form is also associated with the leukemia and the carcinoma. Now the second one is the E-cadherin, and to understand this, let us first understand the process of the beta-catenin and the pathways linked to it. So let me draw a whiteboard again. So one thing to understand is that the beta-catenin plays the central role, and the beta-catenin, if activated, if increased, would lead to transcription activation, and that would lead to cell cycle to proceed. But now who regulates the beta-catenin? Okay, so we have three things to be kept in mind. One is obviously the E-cadherin. So what E-cadherin do is that E-cadherin is a normal protein that keeps when the cells are in close contact, it causes inhibition of the transcription by holding the beta-catenin. So it actually sequesters, or we can say it doesn't allow the beta-catenin to become free, and if it doesn't allow the beta-catenin to be free, then it will obviously cause the cell cycle to arrest or to do not occur. Okay, so the beta catenin would decrease in this when the E catenins are there present, and the cell cycle would not proceed. Now the second mechanism is that by the APC, the APC stands for the adenomatous polyposis coli, and the APC, what APC does is that it destroys the beta catenin. Normally it destroys the beta catenin when there are no signals from the WNT, which we are going to discuss in a moment. So APC destroys the beta catenin, and again the beta catenin would decrease, and that would again lead to a cell cycle to stop or not to occur. Now there is the third mechanism, that is, or not the mechanism, but the part of the APC pathway. Now what this WNT signal does is that WNT signal causes the APC activity to decrease. So the APC activity decreases, and the APC normally causes uh, the deactivation or the catalysis of the beta catenin. So beta catenin would rather increase if the APC activity decrease, and this would cause the cell cycle to proceed because of the increased transcription. Okay, so this is the mechanism of controlling the beta catenin. But now we need we have to just see what is in the E-cadherin, because we have included in the table. So the E-cadherin, what it does is, again I'm stating that E-cadherin normally holds the beta catenin when the cells, this is the beta catenin molecule, so the beta catenin is held by the E-cadherin. This is our E-cadherin, and it prevents the beta catenin to increase the transcription by moving to the nucleus. So that's our main concept over here. So what happens, we can see, is that when the E-cadherin fails, there is no contact inhibition. The contact inhibition is not present, and beta catenin, which is bound normally to the E-cadherin, would be lost because of the E-cadherin activity is lost, and the beta catenin would increase the transcription that would cause the cancer to occur. Now the familial gastric carcinoma is associated with the E-cadherin activity. Okay, the next thing to understand is the Wilms tumor, and recall that the Wilms tumor is a type of mixed tumor, that is, it would have more than one neoplastic cell type. Now, what is a normal role of the WT1 gene? Okay, the normal role is the mesenchymal to epithelial transition, and that occurs in the renal and the gonadial tissues, and that helps in the differentiation. Okay, but now what happens when the Wilms tumor activity decreases or there is some deficiency? That would cause a decreased mesenchymal to epithelial transition, and that in turn would cause more mesenchymal tissue, and that would predispose to the pediatric renal carcinoma. And sometimes we know that the excess of Wilm tumor also causes leukemia. Okay, so there must be a balance, not the deficiency also or not the overexpression also. Both of them cause defects. The next is the von Hippel-Lindau gene, VHL gene, famous for the VHL syndrome. Now, for understanding the mechanism, let's first draw the understand it in the whiteboard. Now, there is something called angiogenesis, and this angiogenesis is important for metastasis. You can think of it easily over here, that when there is blood flow to the tumor, there is a good amount of nutrients reaching, so there would be increased growth, and also some tumor cells may also enter the bloodstream and they may spread. So this thing should be clear, that angiogenesis is helpful for the cancer. Okay, so we must normally decrease the angiogenesis when it is not required. Okay, so now we have something called HIF, that is hypoxia inducible factor, and this HIF-1 alpha, what it does is that it increases when there is oxygen deficiency, that is during the hypoxic states. So now when there is HIF increase, it would cause the increase in the vascular endothelial growth factor to increase. This would promote the angiogenesis, and angiogenesis again would help the cancer to spread or to grow quickly or fast. Now, what governs the activity of HIF? One of them would be the VHL gene, that is the von Hippel-Lindau gene, and this gene, what it does is that actually it negatively regulates the hypoxic inducible factor. So in presence of VHL activity, this would decrease, and the vascular endothelial growth factor would also decrease, and angiogenesis would decrease. And this prevents the cancer spread, or its potential to metastasize. Okay, so let us go back to the table now. So VHL gene, von Hippel-Lindau gene, what it normally does is that it inhibits the HIF1 alpha, and that would inhibit the angiogenesis. Now, what is VHL syndrome? Mainly in the VHL syndrome, we have increased tendency of renal cell carcinoma, but apart from that, we also have cerebellar hemangioblastoma, retinal angioma, and pheochromocytoma that are seen in some cases. Now the next one is the STK11, and for this we need to know what is Warburg effect. We are going to discuss this in more detail in a moment, but for now, what is Warburg effect? Warburg effect is that when the cells are in a multiplicative phase, the cells need some carbon skeleton to have those building blocks to synthesize, and for that they shunt the metabolic intermediates from the mitochondria towards the lipid synthesis or towards some other synthesis apart from synthesizing ATP, so that we are not wasting our carbon skeleton just for burning out and giving the CO2 and making ATP. We want some metabolic intermediate and some ATP. Okay, so this is Warburg effect, and STK11 normally decreases Warburg effect. And if it decreases the Warburg effect, it means there are less metabolic intermediates, or I can say there are less carbon skeleton. And if less carbon skeleton present, then obviously if there are no building blocks, how will the cell multiply? And that prevents the tumor. That prevents the tumor because without these building blocks, the cell cannot make a building or the new cells. Now the familial problems with the STK11 defects are Peutz-Jeghers syndrome, that has increased risk of GI cancer or GI polyps. And the last one on the slide is CDKN1A, and recall that it comes under the P53 pathway, or we can say it upstream to the RB pathway. Normally it causes increased RB expression. And how increase RB expression? By hypophosphorylated state, and therefore uh, if the RB is in hypophosphorylated, then the cell cycle obviously would be inhibited. Okay, and both of the tumors in the familial form are linked to with the RB or P53 presentation. Now what we are going to see is that we are going to see the inhibitors of those cell cycle signaling, who actually inhibits the main growth factor promotion. Okay, so now first of them would be our APC. As I've already stated, that APC forms a part of the WNT signaling pathway, and it destroys the beta catenin in absence of the WNT signaling. Recall that when there is WNT signaling present, the APC is deactivated and the beta catenin is free, and it causes the transcription. Okay. The familial forms of APC, as the name suggests, is linked with the adenomatous polyps or sporadic colon. Sorry, in familial condition, it is linked with the adenomatous polyps, and when present in the sporadic form, it is linked with the colon cancers. The next is the NF1 gene, the one that is involved in the neurofibromatosis type 1 when it is defective. Okay. Now, what normally NF1 gene does? It puts a brake on the RAS signaling pathway. Recall what is RAS. RAS is the proto-oncogene, and obviously if the neurofibromin is decreasing or it is breaking the RAS, then it would decrease the proliferation or decrease the cell cycle. And that's what I've stated. It decreases the mitosis and hence the decrease the proliferation. Now, there are some things that are associated with the neurofibromatosis type 1, and those are neurofibroma, optic nerve glioma, malignant nerve sheath tumors, and not very common clinically, but you may come across this in your examination. The next one is the NF2, the neurofibromatosis type 2, and the important thing is the Merlin. Neurofibromin 2 is also called Merlin, and if it fails, okay, if it fails, then it causes the failure of the contact inhibition, right? And when there is failure of the contact inhibition, it causes the cells to multiply haywire. And what happens when it is defective? Let's see. It has benign bilateral acoustic schwannoma. This is again something that you may come across in your examinations. The next one is the TGF beta, or the SMUD or SMAD. And what TGF beta does is that it is the inhibitor of the proliferation, and hence you can say that it is suppressing the tumor formation. But actually it is more important to understand that which type of cell TGF beta is inhibiting the proliferation. Say if you are saying that the TGF beta is inhibiting the proliferation of our T cell, that is our T cell, that is the lymphocyte, T cell lymphocyte. So that is a problem, because T cell lymphocyte obviously have a role of surveillance in the tissues, and that would cause again the cancer to form, because lack of surveillance would cause the cancer to form. So TGF beta proliferation, it is an inhibitor of the proliferation, but more important is that which tissue it is inhibiting. Okay, if it is inhibiting some normal tissue apart from T cell, then it has a role of help. Otherwise, if it is inhibiting the T cell, then there is a problem. Okay. Now, the sporadic form of the TGF beta defects are associated with the colon cancers or pancreatic cancers, endometrial cancers, and also the stomach cancers. The next one is the P10. The P10, recall we have discussed the PI3K AKT pathway, and there I've also said about the P10, that is, it is the inhibitor of the PI3K AKT pathway, and hence if it is an inhibitor, then it acts as a tumor suppressor gene, and the familial form is linked with the Cowden syndrome. Now there is also one gene, patch1 gene, and patch1 gene causes the unopposed hedgehog signaling that increases the MYC and the cyclin expression. And what the MYC and the cyclin expression, when they are increased, do is that they cause cancer. Okay, because they increase in the proliferation. Hence the mutation in the patch1 would cause a syndrome called Gorlin syndrome. Now, one important concept to understand is that what is Warburg effect? Now, Warburg effect is also called as aerobic glycolysis. It is because glycolysis is occurring even, it is the main thing that occurs even when there is presence of O2. And we are well aware that glycolysis is the main pathway when there is anaerobic conditions. Okay, but Warburg effect is something that is important for the cell proliferation. Okay, and as I've stated earlier, that cells, when they are dividing, they need cellular carbon skeleton and other nitrogen and all those molecules that are required to build as a building blocks. So cells need substrate apart from ATP for multiplications. And now mechanisms for this Warburg effect would be that how basically what we want to do is that we want to decrease the ATP production and shift towards increasing the making of the building blocks. So what we are going to do is that we are going to increase the glycolysis, and that would be by inducing the glycolytic enzymes. Then we are going to increase the lipid synthesis, and we are going to shunt or promote the mitochondrial intermediate shunting. Okay. Now, which pathways are suggest involved, and what pathways induce the Warburg effect? So PI3K pathway is one of them, and receptor tyrosine kinase pathway along with the MYC is also associated with the increased Warburg effect. So because this whole pathway we have already seen in the previous lecture, that MYC and PI3K, all these things cause increase in the cell cycle via the receptor tyrosine kinase. So obviously they should increase the Warburg effect, and by increasing Warburg effect they increase the carbon skeleton, which increases the building blocks, and that causes increase in the cell cycle. Now, to know that Warburg effect is the basis of the PET scan. high yield. What we do is that we inject the radioactive glucose. Now radioactive glucose, since the multiplying cells require more building carbon skeleton, therefore it uses more glucose, but it also saves the glucose, right? Normally the cells would utilize the glucose fully to make the ATP, but those cancer cells would prevent the full utilization of the glucose to prevent the loss of carbon skeleton. Okay, and this is used in PET scan, that those tumors acquire the radioactive glucose and they appear most bright whitish type of color appears in the PET scan. Okay, and last thing to see here is that Warburg effect normally also is present in the cells, and because normal cellular processes also requires building blocks, so a balance is maintained. Okay, a balance is maintained between the Warburg effect and the metabolic requirements of the cell, or we can say there is a balance between the carbon skeleton and the ATP requirements. Okay, so this was the Warburg effect. High to know that it is associated with the PET scan, and high to know that it is important. Warburg effect is necessary for cell proliferation. Another property of the tumor cells is that their ability to escape the apoptosis. Now, apoptosis is the programmed cell death, and the apoptosis has various. The triggers of apoptosis are many. Say we have DNA damage, then the cells, the p53 would detect the DNA damage and cause the uh, apoptosis or senescence. Now we have mutated gene products that would again induce the apoptosis. Now we have some during the embryogenesis of physiological reasons, when there is apoptosis, during say for the clonal proliferation of the B cell, or during the embryogenesis when we need to holoify the our GI tract. Now anarchis is something we need to know, that it is the loss of the basement membrane adhesion with the cells, and this in turn again causes apoptosis. Now there are two pathways of apoptosis, as you may recall: the extrinsic pathway, or also called the Fas ligand pathway, or the death receptor pathway. And the second one is the intrinsic mitochondrial pathway, that is based on the DNA damage. When the DNA is damaged, the cell usually undergo apoptosis by the intrinsic mitochondrial pathway. Now mechanisms of the escape, how the cells escape from apoptosis. So one of them would be obviously the BCL2 overexpression. What is BCL2? BCL2 says B cell lymphoma, B for B cell lymphoma. So it is a overexpression of the BCL2, since it is an anti-apoptotic gene. Anti-apoptosis means it prevents the apoptosis, and hence it gives the immortality to the cancer cells, and it is therefore associated with the B cell lymphoma. The second thing is that we have something called 14-18 translocation. Now normally what is there is that we have this IgH type of the gene, IgH, which is the gene heavy chain immunoglobulin heavy chain, that is highly proliferative, and this is present in the 14th chromosome. But we have another gene we already know, that BCL2. BCL2 is present in the 18th chromosome, and what happens when it is translocated is that there is when the BCL2 comes to this locus, we know that IgH2 is very active, and therefore the BCL2 becomes highly active, and therefore again it if the anti-apoptotic gene is more active, then it would obviously give immortality, and that causes the follicular lymphoma. Now, P53 mediates the activation of the BAX and the BAC. These are the pro-apoptotic genes, and say if we have the problems in the P53, that is, it is unable to induce the effect to the BAX and BAC, so that would also help the cancer to evade the apoptosis. Now, there is something called flip protein that which which inhibits the exchanging pathway. So if there is overexpression of the flip protein, then that would cause the again evasion from the apoptosis. Okay. Now, angiogenesis. Okay, one of the important thing about angiogenesis is that we know that blood favors growth and blood favors metastasis. Blood brings in the nutrients, and blood also takes few cells along with it to some new locations wherever the cancer is going to spread. But what is the mechanism behind the angiogenesis? Hmm. Why angiogenesis occurs? First of all, whenever there are newly synthesized endothelial cells, they secrete the factors like insulin like growth factor IGF or platelet derived growth factor PDGF. And the angiogenic signals are because of the vascular endothelial growth factor or fibroblastic growth factor, very high to know VEGF, and the second high to know is FGF. Then we have angiogenic inhibitors, angiostatin and endostatin. So uh, in short, we have angiogenic signals, the two vascular endothelial growth factor and fibroblast growth factor FGF, and inhibitors angiostatin and endostatin. Now, who are the regulators of these of the angiogenesis? So as I've already said, about one of them is hypoxia inducible factor 1 alpha. Second is the P53. Third, the RAS MPK pathway, or we can say RAS or MYC. The whole pathway is uh, one way or other is involved. Okay. Now, I have already told you about the hypoxia inducible factor 1 alpha, that it is expressed more when there is less O2 tension, or in case of the hypoxia, it is a transcription factor, and it causes the release of the vascular endothelial growth factor when it is when the HIF1 alpha activity increases. Okay, so and it is also inhibited by the VHL gene, and therefore VHL gene inhibits the angiogenesis. Recall. Now, P53 mediates its uh, anti-angiogenic role by using the thrombospondin 1, and hence this thrombospondin 1 causes the angiogenic like inhibitor activity. Now, the RAS, MAPK, or the MYC genes, all of them cause increase in the vascular endothelial growth factor, and hence they give the angiogenic signals. Okay. Now there is a drug which is hard to know because it ties to pharmacology, that bevacizumab. Bevacizumab is one that neutralizes the activity of vascular endothelial growth factor, and hence it is in the trials that is used to decrease the angiogenesis. Now, one last thing to understand is that angiogenesis may also be induced by chronic inflammation. When the inflammation is very high, or uh, when it is for a greater amount of time, that is, of course, it is chronic. Uh, because of the remodeling of the tissues and all, there is increased angiogenesis, and therefore the angiogenesis may occur as a result of chronic inflammation also. The next thing whose mechanisms we must know is very important: invasion and metastasis. Now, there are two main steps of this. First of all, there must be invasion of the extracellular matrix, and then as it enters the blood, there is the vascular spread. Now, what happens, I have just drawn a small flow chart over here, is that we have first loss of the cell to cell adhesion, because the tumor cell must detach itself from the other cells. Once it has detached from other cells, it let me draw something over here. Let's say these are two cells, and this has detached itself from the other cells, so loss of cell to cell adhesion. Now there is some basement membrane over here which this tumor cell has crossed by degrading the basement membrane. Now there are some receptor or proteins which this tumor cell has attached to, and after it goes enters the bloodstream, and then it moves into the bloodstream, and then it moves to some new location, that is the vascular dissemination. Okay. Now let's see each of them one by one. So there is loss of adhesion. So recall the adhesion is because of the E-cadherin. One of the important one is E-cadherin, and the loss of adhesion because of the E-cadherin activity loss. is linked with the adenocarcinoma of stomach, colon, and breast. Secondly, the tumor cell, after losing the adhesion, must degrade the basement membrane. And for degrading, obviously, we would use the proteases or the proteolytic enzymes that are secreted from two sources, the tumor parenchyma itself or the stromal cells. Okay, when the parenchyma, all of them basically secrete same proteases, and well, the important one of them is MMP, the matrix metalloproteinase. Recall that this protein is also used during the tissue remodeling, during the tissue repair, and it is a part of the chronic inflammation also. So chronic inflammation may also be associated with the tumor metastasis. Okay, so the another one is the cathepsin, urokinase, and the plasminogen also. Now it is just for the sake of completeness, you must know that MMP also stimulate the release of vascular endothelial growth factors. So once the basement membrane is broken, we can say the angiogenesis may also be stimulated because of the increase in the vascular endothelial growth factor. Okay. The next step would be the attachment to the receptor and or the proteins, and you may be familiar with most of them, that is the integrins, laminins, or the collagen. That all these proteins normally maintain a differentiated state of the cell, but when the tumor proteins attach to these proteins, there are range of errors. Okay. The last step is the vascular dissemination. And what happens in the vascular dissemination is that one of the fear for a tumor cell is that it might undergo destruction while in transit in the vasculature, and that may be by the apoptosis as a result of anoikis or the host defense system, obviously. Now, while in transit, it may activate the coagulation factors or may adhere to the platelet and may also form emboli in some cases. Okay. Now, as I've mentioned earlier, that stromal cell may also be involved in promoting the invasion and metastasis. And one of the example to be aware of is that hepatocyte growth factor, which is elevated in case of the highly invasive glioblastoma. Now, what determines the site of metastasis? Uh, it is the anatomical location of the secondary metastasis, that is, where are the cells more preferred. If the tumor, say, is of the lung, then it may spread via the roots into the systemic circuits and to the adrenals or to the brain. What is the vascular drainage of the primary tumor? Again, you can just understand with the example of the lung. And it may not be very hard to imagine that there are deposits of the uh, from if there are some GI tumors, then they may deposit into the liver. Secondary deposit may be in the liver. Then we have the environment or metastasized tissue, that is the tropism. This thing is called tropism, and the best example for this is the breast cancer. Now, what is this tropism? Now, certain cells find it easier when they are present in some tissue environment which is favorable for their growth. Okay, and the breast cancer, the genes that are linked with the breast cancer is that they have CXCR and CCR7 receptors. These are the chemokine receptors that some cancer cells express, and this is important in determining their location of metastasis. So both these factors determine where the breast cancer is going to metastasize. Okay. Now, one of the important thing to keep in mind is that the tumor cells rarely metastasize to the skeletal muscle and spleen. Although they are highly vascular, still they are rarely the tumor cells metastasize. There are some preferred sites of the metastasis for some cancers that are high to be aware of, and those are prostate carcinoma, which commonly spreads to the bone, the bronchogenic carcinoma, which is most commonly spread to adrenal and also to the brain. The neuroblastoma commonly spreads to the liver and or to the bones. Now, one of the examples of the tropism may also be that the breast cancer cells, those that arrive to the bone, may themselves make the environment more suitable for their survival. And how they do this is by secreting PTHrP, that is the parathyroid hormone related protein. So it mimics the activity of the parathyroid hormone, and what you can imagine is that it will increase the calcium concentration, and to increase the calcium concentration of the blood, what it should do, it should do the increase osteoclastic activity. And how it increases the osteoclastic activity is by something increasing called rank L. So PTHrP increases the rank L and increases the osteoclastic activity, and in turn it causes increase in the calcium concentration in the blood. Ultimately, it leads to osteoclastic activity increase and bone defects. So this kind of defect helps the tumor cell to survive in the metastasized location. Now, what are the genes that impart the metastatic potentials? We know that E-cadherin is involved to decrease for loss of the cell-to-cell adhesion, but there are some transcription repressor for the E-cadherins also, and those are snail and twist that repress the E-cadherin. And if they repress the E-cadherin, you can say that there is activation. There is activation of the beta catenin and increased transcription, and hence increase in uh, the cell proliferation, but also increase in the metastasis because of the loss of cell-to-cell contact. And now uh, I mentioned over here is that the E-cadherin are downregulated, but at the same time vimentin and actin are upregulated. Now, what are vimentin and actin? The vimentin and actin are actually they are the markers for the mesenchymal tissue, and you can say that these, if they are markers for the mesenchymal tissue, and E-cadherin if it is marker for the epithelial tissue, so we can say there is something called epithelial to mesenchymal transition going on. So when there is metastatic potential, we can say there is something called epithelial to mesenchymal transition going on. Again, the chronic inflammatory states increase the TNF and EGF that may help in metastasis and invasion. The next property to be aware of is that cancer cells escape the host defense, and that is necessary because if the host defenses kill the tumor cell, then what's the point of tumor? Okay, now the main theory behind this is that there is Darwinian selection. What is Darwinian selection? Because of the tons and tons of divisions, there are tons and tons of mutations that accumulate, and those cancer cells are selected that are resistant to the attack or killing by the host defenses. Now, normally the cell-mediated immunity has the major role in the anti-tumor mechanisms, and CD8 positive, that is our cytotoxic T cell, protects us from the Epstein-Barr virus or human papilloma virus induced tumors. Last thing is that NK cells, natural killer cells, and macrophages. The macrophages via the IFN gamma, it has the anti-tumor role. So we have seen the normal mechanisms, but what about the escape? How the tumor cells escape the this our host defenses? So what happens is that the tumor cells lack some antigenicity, again because of some mutations that are because of some Darwinian selection. There is lack of antigenicity, and without antigenicity, we know that if our cells don't recognize the antigen, if there is a lack of stimulation of the immunity or lack of stimulation of the host defenses, then there won't be detection and no killing would there. Now there is reduced expression of MHC molecule, and that too again would help in escape because MHC molecules are required for the T cell activation, and if the T cell are not activated, if the antigen is not presented by MHC molecule, then there is no point in detecting the tumor because they won't recognize, the T cells won't recognize the antigen without MHC molecules. The third mechanism is via the activation of the immune response inhibitors. Now, important to know that CTLA is the part of the co-stimulatory molecule inhibitor. Uh, what CTLA4 does is that it is the inhibitor of the co-stimulation. Normally, whenever there is activation of a T cell, the T cell requires two stimulation, and the CTLA4 is the inhibitor of the other co-stimulation. Okay, the fourth mechanism would be the suppressed production of the immune cell. Now, this is high yield to know that I've already mentioned that TGF beta causes anti-proliferative activity. But whose anti-proliferative activity? Whether it is tumor cell or it's T cell. So if TGF beta causes anti-proliferation of T cell, then it is decreasing the host defense systems, and therefore it is increasing the tendency or chance of the tumor formation. While if it is causing anti-proliferation of the tumor, then it is helpful for us. Okay, so high to be aware of that TGF beta, where it is, which tissue it is actually causing anti-proliferation. Okay, and once again, the CTLA4 is the inhibitor of the co-stimulation of the T cell, and hence it provides a mechanism of escape. Since our host defenses are important for the anti-proliferative activity, so it is not hard to imagine that the immune deficiency would obviously link to cancer. And one of the famous example for this is that Epstein-Barr virus, which normally causes infectious mononucleosis in normal people, but in immunodeficient, it is linked with B-cell lymphoma or inter-Hodgkin lymphoma. Certain tumor antigens work as the tumor markers, and I've mentioned few of them which are high to be aware of. One of them is oncofetal antigen, which are the CEA, carcinoembryonic antigen, and alpha fetoprotein. The next one are the CA125 and CA199 for the ovarian carcinomas. The CD20 is very important marker for the mature B-cell lymphoma. High yield to know CD20, CA125, uh, carcinoembryonic antigen, and alpha fetoprotein. All of these are important to be aware of. They are the tumor antigens used as tumor markers. Finally, we are going to the last of our mechanisms that are going to be disturbed. Those are the DNA repair defects, and there are three kinds of DNA repair: mismatch repair, nucleotide excision repair, and the recombination repair. Okay, one by one we are going to discuss about the diseases, and I have kept a table for easy understanding. So here is the table, and first one of them is the xeroderma pigmentosum, and that is the DNA repair defect is the nucleotide excision repair. Now, as the name suggests, xeroderma, something related to skin. So they have increased risk of skin cancer because of the UV radiation that causes the cross-linking of the pyrimidine residues. The second one is the Bloom syndrome. Now, the Bloom syndrome is because of the recombination repair defect, and important cause for this is the ionizing radiation. Another thing from the ionizing radiation is the ataxia telangiectasia. The next one, that is also because of the recombination repair defect. And the only difference between Bloom syndrome and the ataxia telangiectasia is their symptoms. What happens in Bloom syndrome is that obviously they have increased cancer risk. All of them would have increased cancer risk. But apart from increased cancer risk, which other thing is there in Bloom syndrome is that they have defective development. That is, they are usually short-statured. And in the ataxia telangiectasia, as the name suggests, ataxia, it means the lack of coordination, cerebral coordination, and those therefore have neural symptoms. Okay. The last one is the Fanconi anemia. Fanconi anemia, it is again because of the recombination repair defect, and that is mainly because of the chemotherapy drugs. And anemia, you can see there is bone marrow aplasia. Now, interestingly, the Fanconi anemia is also associated with the BRCA2 genes. Now, recall that BRCA2 is a familial breast cancer gene. One, I can stop here and ask you that BRCA, is it linked with the sporadic cancers? No, it is not linked with the sporadic cancers. It is highly associated with familial. However, the BRCA is not associated with the sporadic cancers. The last one to be aware of is the hereditary non-polyposis colon cancer, HNPCC, and the defect is the mismatch repair. And as the name suggests, it is associated with the colon carcinoma. Now it is the non-polyp type colon carcinoma. Recall that APC are involved in the polyp type, and APC used to degrade the beta catenin and hence acted as a tumor suppressor gene. Similarly, in the HNPCC, there are family of tumor suppressor gene, and high to be aware of are MSH2 and MLH1. Although this HNPCC is the genes are not fully characterized, and some other genes like BAX and beta catenin and all those are also affected, but mainly the mostly MSH2 and MLH1 germline defects are involved. Now, lastly, I would like you to know that chromosomal errors also lead to cancer. Now, I have included the most important examples over here. Obviously, we are going to discuss these again and a few more examples in when we discuss the systemics later. And one of them is the 922 translocation. Is that it is involved in the CML, chronic myelogenous leukemia, and 922 translocation is also called Philadelphia chromosome positive. So what happens is that there is the BCR-ABL fusion gene, and this fusion gene causes excessive activation of the tyrosine kinase. Now, tyrosine kinase, recall that it may activate the MAPK, it will activate the RAS, and the RAS then will activate PI3K and the MAPK and all, and ultimately leading to the cell proliferation, and hence causes the cancer. The next one is the 1418 translocation, which is involved in the follicular lymphoma, and I've already explained this, that the BCL2 is translocated to the IGH locus, and this causes increased expression of this anti-apoptotic gene, which prevents the cancer cells to undergo apoptosis and hence gives some immortality. Okay, so these were the mechanism for the chromosomal errors that lead to, these were the examples for the chromosomal errors that lead to cancer. With this, guys, we have concluded our this lecture on the molecular mechanism of the carcinogenesis. We are still left with the microbial and the chemical carcinogenesis and some carcinogens, which we will discuss in our next lecture. And up till then, bye. And if you like the video, please like, share, and subscribe. If you have any questions, please put them down in the comments. Bye.